Welcome back, everyone. So we are now in the sixth session of the day, and we will have João Marques and Gábor telling us about low coverage and reduced representation, genomics, merits and limitations. Go ahead.
Hi, Zef. Hi, everyone. Hi, Gábor. So since this morning and now talking about a downstream part of all our analysis. So most of what we'll discuss today is more related with the analysis itself and the case studies, and this falls into having very nice reference genomes, as we discussed through the last sessions. So either me and Gábor, we lead case studies of BGE. So we are part of work package 11, and today we'll basically explain our experience on these case studies and on the development of these case studies. And with this, give examples of the use of low coverage and reduced representation genomics. So I will briefly start with the initial pool, just to have a feeling of the audience. It will be very quick, and then I will pass the word to Gábor that will present two case studies where it is involved, and the use of reduced representation genomics on them, and then I'll be back. So let me just start this sharing the screen. Can you see my Mentimeter screen, right? Nope.
No.
Okay, maybe we will need to stop share before we share it again. Okay, now it's okay?
Yes.
Yeah, okay. So please, if all you can to join the Mentimeter, we'll use it through the presentation. So it would be nice if you could stay in the page. I will give just a few seconds to everybody join. Okay, perhaps I will start. So we'll start briefly with the experience of each of you. So basically, the idea is to order and to rank the sequence strategies which you are familiar with. And again, the idea is just to have a brief overview of all of you. Okay, so I think that Gábor, most of the audience is here for you.
I hope not. Yes. Resequencing is coming up, so.
It is coming up, yeah.
Oh, yes.
Okay, I think we have already a good pool. So we have a diverse experience, which is good. So I'll maybe jump to the second one. And then now is the level of experience. So from nothing to advanced users. And don't be shy, don't just put the intermediate, just to Okay, I think as in the case of the type of methods, we have a very vast audience. So I think that this already gave us some hints on how to proceed. So now I will pass the word to Gábor. So I will stop sharing. And thank you all for participating in the poll. And Gábor, the floor is yours.
Thank you very much. Dear ladies and gentlemen, dear colleagues. So let me introduce you our groups and its work. And as João has already said, I would like to talk about RADseq because our research group is mostly working with RADseq. And actually we have very large, very extensive experience with different organisms. Some of them are unconventional, most of them are more conventional. So in this presentation, I would like to really talk about two case studies, what we do within the framework of BGE. But before I start with that, let me very, very briefly have an overview, give you an incomplete overview of reduced representation genomic approaches. So in these approaches, we are simply trying to restrict our sequencing effort to usually thousands, sometimes 10,000 of genomic locations. And thus we can achieve high vertical coverage. And of course, it comes on the expense of low horizontal coverage. Therefore, we will see a little from the whole genome, but we hope that part, what we see is representative. So there are different main approaches. One is target capture, where we try to pull out genomic regions from the genome. RNA-seq or transcriptomics, when we are focusing only on transcribed genetic information, and there's RADseq, sort of approaches in which we are using restriction enzyme or enzymes to locate anonymous part of the DNA what we sequence. So as I said, I will mostly talk about RADseq here. The result of such an experiment is a low density genome-wide variant discovery. So it's very important to emphasize it will be low density, because as has been said already several times, the horizontal coverage is low, typically 1–2% of the genome what we can see. Sometimes it goes up a little bit higher, but normally it's really a few percentage, but at high read depth, which will give us some confidence for SNP calling and downstream SNP filtering. There are, of course, almost countless number of variants of RADseq now. I'm mentioning here only the three most important ones: single digest RAD, or the original RAD, some call it like this, when we have a single restriction enzyme and there's a randomly shared other part of the fragment what we are targeting, and we use the PCR to amplify fragments with the two adapters what we ligated on. There is the double digest RAD, which is very popular because you don't have to shear your DNA. You don't need to use the sonicator. And then there's basically it was developed as a separate technique, but now it's considered clearly within the family of RADseq, genotype by sequencing, when you simply put the same adapters to the chopped up genome and use the PCR to amplify those short fragments which are with the two adapters. Okay, so what is a typical problem for RADseq is the PCR duplicates, because you cannot avoid PCR duplicates, which come from the step, is the PCR step. And of course, it can cause real problems because it can distort the original allele frequencies. What is a cure for that is to use the original version of RAD, when you have the randomly shared 3' ends, because if you use this original red, you can use post-bioinformatic filtering for the PCR duplicates. The other blends of red doesn't allow for this. Actually, this is the reason why in our lab we are preferring to use SD red, so the single digest red, although it's somehow technically more challenging because there are much more wet lab steps. So after this very big, very arbitrary overview, let me come to the exact showcases. So we are working with 2 mammals in the frame of BGE. One is the critically endangered European hamster, and the other one is the endangered Hungarian birch mouse. And in these animals, we used RADseq to have an overview and get a picture about their genetic structure. So very briefly about these animals. European hamster has a very large distribution area, but because of a steep decline on all of this very large distribution area, it was recently considered to be critically endangered by IUCN. So we started to put more and more efforts into the understanding of the genetics of these species. And actually our Russian colleagues did recently very much, but they only used genetic approaches, so candidate genes usually. And what is something interesting, what is going on all across this large distribution area is the urbanization of the species. Sometimes the species invades villages, sometimes park areas in cities, and the population simply starts to live there and disappears from the neighboring agricultural land where it used to live before. And this is something very interesting in our view and can have a conservational implication. Therefore, we are asking several questions, but one of the most important questions about this species is this urbanization, whether it has any mark on the genome or how this urbanization is going on, whether this can be used for long-term conservation of the species. Of course, these questions we would like to answer with dense SNPs and not low density SNPs. So we will do some resequencing, but before going to resequence blindly some individuals, we wanted to have an overview of the genetic structure of the species, at least in our region. So we made a first RAD library where we did de novo RAD locus assembly. And actually this has shown a very nice picture to us where the Danube, the most important river of the Pannonian Plain, was found to be an important barrier because this is the basic split within the populations or the groups. And but the number of samples were not so high, so we managed to obtain samples from Vienna, actually an urban population, more samples from Austria, Czech Republic, Serbia, so basically the neighboring countries, and we were also being sent some Russian samples that represent an eastern lineage of the species. And altogether, we analyzed 92 samples from 2 libraries, so there were 2 libraries. I managed to use this time the reference genome, which is under construction in Spain. And without going into the details, we had quite a lot of SNPs, what we could analyze for this work. And again, some practical information or something which is, I think, very important to consider when we are using RADseq, and especially if we are combining libraries. We always include technical replicates, which are simply the repetition of the same sample in the second library. And after the bioinformatic work, they should come together as a monophyletic unit very close to each other, or as close as possible. And I think this is something what we need to consider when we are designing a RADseq experiment, because honestly saying, there are so many loci, so many things are going on. This is a good way, in my view, to somehow be confident in what we are seeing. We also had some troubles with other species with chimeric reads, and this can happen easily in RADseq when the adapter is somehow incorporated into so the index barcode is incorporated into the target fragment, the insert, and then the samples will cluster according to index barcode. And to avoid this, we just simply name our samples according to the barcodes and check whether there's any evident clustering in the results. Luckily, with the European hamster, we didn't have anything like that. I think these chimeric reads are very much problematic in RADseq. And in some libraries, we had to drop all the data into the bin because of this. Something what should be studied later. Okay, so these are the results for the Pannonian lineage of the European hamster. A very clear picture in my view. There's an interesting sample here in the base of the tree. Of course, the Russian samples are light years away from the Pannonian samples. I think it's absolutely normal. And then there is this soil sample from Transylvania, the Transylvanian basin. We don't know why it is so separate. And then we have 1, 2, 3 main lineages. And these 3 main lineages are clearly connected to the Danube, because we have one lineage north of the Danube from Czech Republic and northern Austria. And just across the Danube, the Vienna samples are here in this lineage, where we have samples together on the southern side of the Danube. And then the rest is in this group, which is east of the Danube. So basically, the Danube is really an important barrier for this species. And if we compare it to the previous results, the fine details are also the same, just the additional samples simply find their place in the phylogenetic space. And the story is something like that, what I sketched onto this map. The thing is, for our main goal, which is selecting samples by knowing the genetic structure for the resequencing experiment, I think we can identify 2 clades and 1 grade. And within this, there are some populations which urbanized, some which are still living in the agricultural environment. And we can pick from those individuals what we will resequence. And actually, with this genetic structure in the background, we could make an informed decision. And one way of having an informed decision was also to check kinship between the samples. Of course, because you are sampling these individuals on the field, you don't know whether they are close relatives or not. They will not look nice in a population genomic analysis, especially when you do some comparative genomics. Therefore, we wanted to avoid some closely related individuals. And from this heat map, you can see there are some individuals which are closely related. And what we did, we simply excluded the left part of this bar chart, which shows the more closely related individuals, and picked the individuals from the right side, which are less closely related. And this is our choice. So the yellow highlighted individuals will be sent for resequencing. And of course, 18 individuals will be from the agricultural environment, 18 individuals will be from urban environment, and the 3 main genetic groups will also be represented. So that was the main goal here. We had an additional goal of looking at genetic diversity. Genetic diversity assessment using reduced representation is especially challenging because you simply exclude invariable positions, and therefore that will bias observed heterozygosity estimates. For those who would like to learn about this, I recommend this very nice paper from 2021. And because of that, it's almost impossible to compare the results of reduced representation genetic diversity assessment between studies. But I believe within the same study and within the same library, we can assess the genetic diversity of the populations compared to each other. And in this respect, we didn't see any apparent differences between the urbanized and the agricultural populations. Somehow the Russian population shows lower genome-wide heterozygosity, but it is definitely a very different clade, so anything can be at the background here. So very briefly about the second part of the story is the Hungarian birch mouse, which is part of this Cicista subtilis species group that really goes through the steppe zone. And the westernmost member is the Hungarian species that used to be once very widespread in the Pannonian Basin, but now it only occurs in this roughly 10 square kilometer territory. And within this territory, we have some subpopulations which are only few kilometers from each other. We don't know whether these are really subpopulations or these are just places of fragrancy. We don't really know anything about the species. So here we wanted to use first microsatellites, and we devised new microsatellites, but they didn't really give any meaningful resolution. You can see that only 2 genetic groups were identified by structure, and there is no apparent structure within the neighbor joining tree itself. So we did some RADseq. It was mapped to an ad hoc genome, and RADseq provided much more detailed resolution. As you can see, all the subpopulation, all the samples from the same subpopulation formed a clade. So therefore, yeah, and of course we were able to use the genomic data to see within this very fine and small territory some genetic clusters, although these are definitely suboptimal clusterings, because even with the genomic method, only one population was assessed as ancestral population here. So the thing is, RADseq is certainly a significant improvement in phylogenetic resolution compared to microsatellites. And actually, we also have another study which was published in 2021. So certainly, microsats are absolutely super. They are very easy to use, work with, and they are comparable with other studies. But the phylogenetic resolution is much higher with RADseq. Okay, and at the very end of this presentation, I collected some advantages, what I believe are advantages of RADseq. Large number of samples we can include. Actually, it's a very fast assessment of genetic structure. We can tackle PCR duplicates if we go for the original RAD. Of course, the phylogenetic resolution will be much higher than any genetic approaches. Of course, if we have a reference genome, that will help orthology assessment greatly. Nevertheless, there are problems. There are always problems. I think it's a very big problem that there is no standardized way of measuring genetic diversity with RADseq and reduced representation in general. Chimeric reads can make problems. PCR duplicates can really bias the allele frequencies. Of course, if we don't have enough read depth, that can spoil anything, but I think it works everywhere in next generation sequencing based genomics. And of course, paralogy can be a serious issue when especially when we have to de novo reconstruct the RAD loci. So this is the end of my talk. I give my acknowledgement for all the co-authors, and I stop sharing my screen because I think it's better to move on, and I give back the word to João. Oh.
You, Gábor. I think you can already see my screen, right?
I cannot stop it. Yeah, there we go. Thank you.
Okay. So briefly, I will change a bit the way I will present the case study where I am involved and put the effort more on the rationale that drive us to use low cover genomics in the end. So very briefly, our case study is about myxomatosis, a virus on the Iberian hair. So it's in the beginning was a clearly for us at least case control scenario, and I will explain why. So we have an original naive natural population of hares in Iberia that suddenly face a new virus. This virus came from the rabbit, and what people start to see in the field was some individuals look resistant to the virus and others susceptible. And so when we designed the case study, was simply compare the resistant ones with the susceptible, but we know that these kind of things are much more complicated. And so we start to discuss this with people in the field, and apparently the virus does not affect other species in Iberia. And we also know from previous work that there is historical gene flow between the species. And this is just to do a brief overview of the situation. And so when we start to discuss with experts in different areas, and that's the important message, that we should involve stakeholders in our case study design immediately from the beginning, is that for us, what we looked for as a simple case control scenario in the beginning was much more complex in the end. And so we end up with several questions. One is it is really host resistant in this case and is genetically determined? And if yes, it's linked somehow with this historical gene flow between species. But we could see it in the side of the virus itself, so it could be related with virus variability, and with COVID, we all saw what different strains of the virus did. But it can also be related with many other factors like body condition, age, co-infections, or it can be even more complicated and a combination of different aspects. So the thing that we came up in the end was, okay, how can we properly test these scenarios? So in relation with sampling and sequencing designs, and of course, considering the money that we had available. So in relation with the sampling scheme was quite easy to define, I would say. So as a GWAS study or case control study, we think that we needed a very good number of samples. So at least we came in the end to a number of 100 individuals per condition. So 100 susceptible and 100 resistant. And for that is already a lot of work, because all the individuals needs to be tested against antibodies, and and knowing the presence of the virus or not, and doing a lot of observations related with the lesions, the body condition, etc. So we collect all this information and we have all this information. But then the question was, okay, and now what about the sequence scheme? And of course, the ideal scenario for this kind of cases is just shoot it with the whole genome, high coverage data for each individual. So this will allow us to individual variation, so to pinpoint each individual in the population or in the data set. It would allow us to also check for virus variability because it's a DNA virus. We could infer very high confidence individual genotypes. We can even detect some DNA-based co-infectants from the individual, but of course, this has some problems, namely a very high cost just for sequencing is more than 1,000, and I'm taking out the human resources that we would need to do that, and of course demanding computational resources. So at least 12 terabytes of raw data plus the time that this will need to be analyzed. So impossible. So what are the alternatives? And as we saw on Gábor talk, one alternative could be a reduced representation genomic approach, although this will not enable us to neither to access to virals and DNA-based co-infectants, but would also not enable us to do a genome scan analysis, so analyze all parts of the genome. But the advantage, of course, as Gábor told you, is low cost and as a nice time for computational analysis. Well, so it takes a short time to analyze in comparison with high covered genome data, and doesn't need a lot of space for storage as well. But as I told you, don't cover most of the attributes that we would like to analyze. So another option could be full sequencing. So full sequencing allows us to do genome scans, but on the other hand, we cannot pinpoint each individual variation. We cannot analyze the virus variability, and we cannot also infer genotypes, individual genotypes. So also not an option. So for us, the only option that could fit all this would be low coverage, whole genome resequencing with individual barcoded. So it means that all the individuals that we put on the sequencing on the library are individual barcoded. So we can recover this. And although we have a very shallow sequencing for each individual, like 2 to 3x, this allows us access to virus variability, individual variation, although we know that we cannot infer individual genotypes, at least with high confidence, and we can also access to some co-infectants. So this provides us with a medium cost solution, and also with the data set that we can manage in time, so in the time of the case study. The problem, as I told you, is that this does not allow for a correct inference of individual genotypes. So it makes the analysis much more complex because most of the methods that are available are genotype dependent. And so we need to adapt to, well, most of the analysis will be done as in the case of the normal pool-seq. But in the end, we can somehow recover the individual information to confirm some signs. And I think that now it's time. Well, we have 10 minutes, so I think it's better to speed up things and go to do a brief pool to try to understand what are the problems of and the limitations that most of you saw on on both methods and from your previous experience, and perhaps reserve some time to discuss, although I'm seeing that there are some discussion on the chat, so thank you to all that are participating on that nice discussion. And so let's back to Mentimeter. Okay. And now I need to be very quick in most of this analysis, so please be as fast as possible. So, question number 1. I know that this bit does not allow to multiple answers, so I would say that you should choose the one that you use most.
So really here people have a lot of experience with reduced representation genomics. Oh, no.
No,
now
it's 50-50.
Yeah.
Okay, and so I will ask from those that use reduced representation genomics, what method in particular? It's really challenging to deal with the Zoom bar at the same time as all the other screens moving. Okay, so most on RADseq, which makes sense, and I would say that was expected. And now, what was the primary reason for choosing? Gábor, do you want to comment the results? Because I think that
Yes, I think
all this.
Yeah. I'm at the same time answering the questions in the chat. So the thing is, basically, it's certainly very cost effective. And I think I read somewhere the RADseq is the poor man's genomic approach. And yeah, it's relatively cheap compared to even low coverage resequencing, I think.
So you think that if we link with this morning gap analysis section that talked about inequalities between widening countries versus developed ones, versus the rich guys, do you think that RADseq is mostly used on widening countries?
Yes, absolutely. In our country, in Hungary, the resources are much more limited, and I really need to fight very much for those 3,000 euros what I spend on one library. So But I cannot say this is something what is impossible to get, but not easy. So honestly saying, it's part of
Yeah,
the story.
the cost, yeah. Okay, and moving on to the major limitation.
Yeah, it's interesting. Most of you are thinking a lot of important genomic regions are lost because there were a very fierce argument between those who were pro and against RADseq. And the final conclusion was, or actually a lot of researchers were clearly arguing for the representativeness of RADseq. And if I think of my own experience with it, we really managed to find some very nice adaptive regions and a lot of information from RADseq, although it's really very low horizontal coverage, but somehow it really finds almost all parts of the genome. And we also used RADseq for identifying sex markers in amphibians. So basically that's also tells me that a lot of parts of the genome is still represented. Therefore, I wouldn't be much concerned about the loss of important genomic regions, but it seems the community thinks the other way around.
Yeah, well, I don't know if it's because a lot of guys that don't work on reduced representation genomics um, did answer as well. So in comparison, they will say that is the biggest advantage, because I would think that for the ones that use it regularly, I would say that well, the difficulty on the analysis will pop out as the most important and most challenging aspects. But okay, I think that we should move because we need to do the same for the low coverage as well, and we have 2 minutes left. It needs to be even faster, although here it's a bit easy, I would say, less options. Okay, so here was quite unexpected for me because by heart, I would say that a lot has been done on tool sequencing on the last years, but apparently not as much as I think. Because the importance of the cost here is on the other way around, right? Because the sequencing itself is the same, so the cost with the sequencing is the same, but the cost with the library is several times bigger. So for example, in the case study that I just presented is 200 times bigger than in the case of low coverage versus tool-seq. So
I suppose a lot of researchers are discouraged because you lost the information of the individual, and probably this is why people don't really like to use it. Sometimes it's good to know about the individuals who you studied. Probably this is the reason.
Yeah, I agree. And perhaps is that thing that if you have money, you will go for the most informative one. And if you don't have money, probably you will go to reduce representation techniques instead of doing tool-seq. Okay, so this comes to the primary reason. Perhaps this will give us an hint. So money seems to play the role everywhere. Was already in the morning and here in the afternoon. So
Money or because in the deep we are all ecologists and we are interested in populations and population level variability.
yeah. Okay, and finally, just because we are really on top of time. Okay, there is a typo in the limited ability to recover individual genotypes. Sorry for that. And perhaps that's one of the reasons that it was so low in the. Okay, thank you all for the participation. Yeah, and yeah, the variant calling is a major issue, and still the reason why, if we have money, we'll go for whole genome high coverage sequencing. Okay. Okay, I think that we are finishing our time. I'll just leave you with a compilation of everything that we discussed here today. Thank you for all that participate. Thank you, Gábor, for hosting the session, and thank you to the organization. I think that we'll come back in, well, now 12 minutes for the next section.
Yes, thank you, João. Thank you, Gábor, for a very interesting presentation and discussion. Yes, in the end, I think everyone always needs to be aware of the objective of the application or research that they're doing and the limitation of each approach. And then it's always strike a balance between what we're able to collect and be aware of the limitations of the...
To collect and to analyze.
Yes, and be aware of the limitations of depending on the initial objectives. But that's what we struggle every day. Okay, thank you, everyone. So as João said, we will be back now at 4 with the final session of the day devoted to temporal analysis and historical sampling.